Ever pondered on what a virus looks like at a molecular level? Viruses, despite their simplicity, are truly fascinating. They consist of an extracellular infectious stage known as a virion, which generally comprises a viral genome encased in a protein coat, or capsid. Some virions even sport an envelope, a stylish accessory borrowed from the host's plasma membrane. At the heart of a virion lies its genome, the nucleic acid, DNA or RNA. Interestingly, a virion contains only one type of nucleic acid. So we have RNA viruses and DNA viruses, but no hybrids. The size of these viral genomes can vary widely, ranging from a modest 3,000 base pairs to a whopping 1.2 million base pairs. Now, the structure of the nucleic acid can be either single-stranded or double-stranded. Yes, we have double-stranded RNA and single-stranded DNA viruses as well. The DNA can be linear or circular, and a virion may contain a single strand of RNA or several segments. The RNA viruses come in two flavors: positive sense and negative sense. Positive sense RNA has a 5:3 RNA genome, which can be directly translated into proteins, while a negative sense RNA, with its 3:5 RNA genome, needs a little help. It must first be transcribed to positive sense RNA before it can be translated into proteins. Now imagine a safeguard for this genetic material, the capsid. Picture a knight suit of armor designed to protect the knight from harm. That's what the capsid is to the virus. This protective shell is made up of proteins and shields the precious genetic material within from the harsh external environment. Let's delve into the structure of this protective shell a bit more. The capsid isn't just a simple shell, it is a complex polymeric structure. It's made up of smaller units like building blocks known as capsomers. Each of these capsomers is constructed from different types of proteins, but where do these proteins come from? They're actually encoded by the virus's own genome. This is where the host cell comes into play. The capsid proteins are translated inside the host cells using the host's own protein synthesizing machinery. It's a bit like a Trojan horse scenario, where the virus uses the host's own resources to build its protective shell. Once the capsid is constructed, it encloses the viral genome, forming what is known as the nucleocapsid. This structure serves two crucial roles. First, it protects the viral genome from physical and chemical stresses. Think of it as a sturdy fortress, safeguarding the valuable treasure within. The second function of the capsid is equally important. It contains specific attachment sites that allow the virus to adhere to the host cell. It's like a key that fits perfectly into the host cell's lock, allowing the virus to gain entry. Once the virus has attached itself to the host cell, the capsid aids in penetrating the cell membrane, facilitating the invasion of the host cell by the virus. To sum up, the capsid is more than just a shell. It's a protective armor, a key, and a vehicle all rolled into one. It plays a pivotal role in the survival and proliferation of the virus, safeguarding the viral genome and enabling the virus to invade host cells. Hence, the capsid acts as the knight in shining armor, protecting the precious genetic material within. Ever thought of a virus with an outerwear? Meet the envelope. You see, not all viruses are created equal. Some come with an additional layer, something akin to a protective coat, known as an envelope. This envelope is derived from the host's plasma membrane. Yes, the very cells the virus invades donate this outer layer. But what exactly is this envelope made of? Well, the envelope is essentially a lipid bilayer membrane. If you recall from your biology lessons, a lipid bilayer is a universal component of all cell membranes. Its main function is to act as a barrier to keep ions, proteins, and other molecules where they need to be. Now, the viral envelope doesn't just borrow the host's membrane, it adds its own touch. Inserted into this lipid bilayer are virus-specific proteins, or glycosylated membrane proteins. These proteins are encoded by the virus itself and play a crucial role in the virus's life cycle. Think of these virus-specific proteins as the virus's personal brand, its unique identifier. They often appear as projections, spikes, or knobs on the surface of the virus. If you've ever seen a microscopic image of a virus, these spikes are what give viruses their crown-like appearance, such as the infamous coronavirus. But these spikes do more than just look menacing. They help the virus attach to host cells, allowing the virus to infect these cells. In essence, these spikes are like keys, and the host cell receptors are the locks. Once a spike finds a compatible receptor, the virus can gain entry into the host cell, beginning the process of infection. Not only do these proteins assist in infection, but they also play a vital role in the virus's survival. They protect the virus from the host's immune system, allowing the virus to reproduce and spread before the host defenses can mount an effective response. So, the envelope serves as the outer shell, providing an additional layer of protection and identity to the virus. This outerwear is more than just a fashion statement. It's a survival strategy, a way for the virus to sneak past host defenses and carry out its mission of replication. To bring it all together, let's summarize the molecular structure of viruses. At the core, we have the heart of the virus, the nucleic acid. Acting as the viral genome, this DNA or RNA blueprint is the instruction manual for viral replication. The nucleic acid can be single or double-stranded and may even vary in its sense orientation. Next, we have the capsid, the protective protein coat encasing the viral genome. Formed from structural subunits known as capsomers, the capsid serves not just as a shield but also a vehicle for attachment and penetration into host cells. Finally, we encounter the envelope, a lipid bilayer membrane that cloaks some viruses. Notably, this envelope is borrowed from the host's own plasma membrane, but it also showcases virus-specific proteins, often seen as spikes or knobs. In conclusion, viruses, despite their simplicity, are incredibly complex in their structure and functions, all thanks to their three key components.